So hello everyone. While the last ones keep arriving, just give a brief introduction to this webinar series. So these are our PlasPhen webinars. These are seminars that come from our group, which is called GDR Plasticité Phénotypique. It's a network that we started in France, where we work on. We have several people working on many aspects of phenotypic plasticity. And last year we started some more international webinars where we have the privilege to host very nice speakers. As you will hear today, we are not going down in quality from the previous one, so it's really great. And so just to tell you that this is a webinar mode of Zoom. So you can type your questions at the bottom during the talk in the Q&A or QAR tab, which is different from the chat tab, which you can also use if you have some technical issues. But you can type your questions there. You can also vote for the questions you would like to see asked. If you have the same question as the person, you can vote for it. And then at the end, we'll ask our speaker these questions, and you'll be able to interact with him and discuss. So with this, I'd like to let Patricia introduce our speaker for today, Huey.
Yes, so good afternoon to all of you. It is with great pleasure that I introduce today's speaker. So he's Huey, Professor Emeritus at the University of Washington in Seattle. Huey began his career doing a thesis under Ernest Edward Williams at the University of Harvard, which he defended in 1975. He then did a postdoc at the University of California at Berkeley, and in 1977, he obtained a position as assistant professor in the Department of Zoology at the University of Washington, where he will spend his entire career. I think I'm okay with until now. So I'm not going to insult you by presenting Ray's work on thermal response curves that we are all familiar with in our small community on phenotypic plasticity. Instead, I will draw your attention to the other keywords you can find on Ray's webpage. Indeed, you will find keywords as diverse as phenotypic plasticity, which is a very good choice, Drosophila evolutionary physiology, excellent choice, lizard ecology and evolutionary biology, well, nobody is perfect, but also mountaineering or paleophysiology, which might seem more original. And in fact, we can say that Ray is a pioneer of a very fashionable word nowadays, which is interdisciplinarity. He's a passionate person, and it is this passion that, I believe, guides his scientific choice. And I had the chance to work with this great man, and I would like to end by underlining his great human qualities, his modesty, despite an impressive CV, his kindness, and his ability to discuss everything with a good glass of wine. So thank you again for agreeing to do this seminar very early for you, and I let you the floor.
Thank you, Patricia. Let me try to share the screen here. Whoops. Okay, is that visible? Great. Well, thank you, Patricia and Luis, for this opportunity. And hello to various friends that I see are on the program. This afternoon, I want to talk about seasonal acclimation studies. It's an aspect of plasticity. And I'm going to take a fairly critical view of the current kinds of protocols that are being used in the field and make some suggestions that I hope will lead to more realistic kinds of patterns. I should say that I became interested in seasonality very early in my career between master's and PhD, 1969 and 1970, had the opportunity to spend a year in the Kalahari Desert of Southern Africa, working with Eric Pianka, studying the ecology of lizards. And we camped out for a year. The Land Rover we had did not have a heater or an air conditioner, so we experienced the seasons full force. And it became obvious to me during that time there that what controlled where and when lizards were active was really the thermal environment and how that changed seasonally. And that formative experience really prompted me to be aware of seasonal biology, and it's important, at least in temperate zone areas. But through most of my career, I actually ignored seasonality. Wrote one paper on Kalahari lizards and a couple on fruit flies. But by and large, for most of my career, I just was working on other things. That changed about 5 or 6 years ago when I reread this paper by Raymond, which was published in Ecology in 1941. Coles was a professor at UCLA. He's famous for really laying out the field of behavioral temperature regulation. But this was an earlier paper, and Coles went to the desert in winter, Southern California, looked at his feet and was surprised to see snake tracks and lizard tracks right in the middle of winter. He became interested in how does an animal that's underground, buried in the sand, how does it know that it's a warm, sunny day and then could come out and take advantage of that? And Cole speculated that, well, it would have to be shallow because when the sun hits the sand early in the morning, the heat pulse travels slowly down through the soil. So if a lizard or a snake is shallow, it's going to get a signal that it's a warm day quickly. But if it's down deep in the soil, it's going to take many hours before that heat wave comes. So there was a clear advantage to being shallow. But Coles also realized that there was a distinct trade-off that an animal that was shallow ran the risk of freezing at night or being detected by a mammalian predator during the day. When I read this, I was stunned. I mean, I'd known about this paper all my life, but I'd clearly never read it, all my academic life, I should say. But here's 1941, Coles is talking about trade-offs. And to my knowledge, this is one of the earliest examples of trade-off thinking in ecology. And I was fascinated by it. I started wondering what's known in the many decades since Coles wrote this paper. Started looking around and found that the answer was basically nothing. The paper and the ideas in it had been overlooked by me and everybody else. So this led to a study with Liang Ma, Ophir Levy, and Michael Carney, published last year. We couldn't find real data, so we simulated it. We took soil temperature data for many sites around the continental United States where we had time series at 5 depths. We could then use those soil temperatures to estimate the physiology of animals subjected to those temperature profiles. So we could look at how cumulative metabolic rate changed over the seasons, how risk of freezing, and so on. But it was all simulation studies. And we were left with a number of unanswered questions about what are lizards doing in the winter, the ones that are active. And that led to a second paper. It came out a couple of months ago, working with Don Miles and Eric Pianka. And this is going back to the Kalahari, the data that we collected 50 years ago, and looking for patterns of activity there. And it was a lot of fun to go back and recycle data from one's youth and still find them relevant. But an issue that remained was the physiological data looking at seasonality, how acclimation shifts affects the physiology and performance of these animals. And that led to the study that I'll be talking about today. It's in collaboration with Lauren Buckley, a colleague at the University of Washington, looking at the available literature on seasonal acclimation patterns and taking a critical look. We can start just with a simple example of why physiological acclimation to seasons might be useful. On the left have animals that are, hypersensitive here, have animals that show no seasonal acclimation, no shift in thermal sensitivity, whereas the ones on the right do. And if an animal can shift its physiology to match or to respond to the seasonal change in body temperatures, that can enhance performance in a seasonal environment. And Monty Slatkin and I years ago developed a simple formula where you can quantify the benefits of acclimation, where if you know how performance or fitness varies as a function of season, function of temperature, and how that varies seasonally, if you know how the distribution of body temperatures vary seasonally, you can then get understand how an acclimation shift enhances performance or fitness. And that general equation has been used now in a number of papers. Seasonal acclimation is relevant to a variety of different biologists. If you're a physiological ecologist, knowing whether physiology is shifting can help you understand whether animals are using plasticity to buffer responses in the environment. If you're an evolutionary physiologist, you might look at whether these shifts are adaptive and model how plasticity is a buffer under climate change, as Luis has done. And if you're a mechanistic physiologist, seasonal acclimation is a really rich opportunity for studying cues and mechanisms. And the vast majority of work that's been done on physiological acclimation is looking at sort of mechanistic sides of things. There are three basic approaches that people have used to study seasonal acclimation. One is simply going to the field, collecting animals, bringing them back into the lab, and measuring some set of traits. A related technique is to set up enclosures in the field, seed those enclosures with animals, and then periodically take them into the lab. But by far the most common approach is to bring animals into the laboratory under controlled conditions that hopefully mimic the conditions that animals would see in nature. And this is the vast majority of studies that have been done. People have been doing this for a very long time. Claude Bernard, 150 years ago, noted that a frog or a toad in summer is a different animal from one in winter. And there, I would guess, tens of thousands of acclimation studies in the literature now. So there's a long, long history. And one would think, given that long history, that the protocols for studying seasonal acclimation would be very well worked out and well validated. And I think that's not the case. There are two classes of problems that one can highlight. The first one is well known. It has been pointed out in the literature many times. And that is that typical acclimation studies that are done in the laboratory use environmental conditions that are not ecologically relevant, or at least not ecologically validated. So people are often guessing how to set up a temperature for summer versus winter. That's well known, but it's still an issue. A second and more subtle point that, as far as I know, has not really been addressed is that an acclimation experiment takes away the behavior of animals. This is a quote that I wrote for a discussion of laboratory natural selection experiments and being critical of them, but I'm going to apply it here to acclimation. When you take an animal and you bring it out of the field and you put it in the lab under fixed conditions, you take away its behavior. And behavior is the primary way that organisms deal with environmental challenges. And I think it's an open question is when you prevent behavior, when you block behavior from being used, are you inducing artifacts into your analyses? I don't know, but I think it's worth addressing. Okay. Just remind you that behavioral temperature regulation is something that's been well studied for many, many decades. And here's just an example, say, of a lizard and a fly. Whoops, you have some measure of performance. These are the temperatures the animals are selecting. So they're avoiding extreme heat or extreme cold, and they're active typically at temperatures that are enhance performance. So the consequences of having behavior are well described. So what I want to do this morning is, or your afternoon, sorry, look at 4 topics. I'm going to concentrate on temperature because that's something I know most about, but I think these are other aspects of setting up an acclimation experiment that are worth thinking about. So let's start with field studies. In principle, it's one can go to the field at different seasons, collect animals and bring them back into the lab. This is probably the gold standard for seasonal acclimation because you're seeing what actually happens to organisms and their physiology in nature. But there are obvious challenges. Finding animals in winter can be challenging. And in some places it's impossible, in others it's dangerous. So, but it is sort of the gold standard, I think. Lauren put together this slide where people working with a variety of different taxa have gone to the field at multiple times during the year and measured heat tolerance or cold tolerance. And you can see that there's seasonal variation. They're more heat tolerant in summer, they're more cold tolerant in winter. Not surprising, but it shows just an example of this type of approach. The challenges here, other than the difficulty of getting animals at some seasons, is that these approaches are purely descriptive. You can do experiments in the field, but these approaches are purely descriptive, and they are specific to the particular animal and to the particular locality, at least to some extent. The variation is to use artificial enclosures. This is an example from work by Pete Zaney working in Oregon, where Pete, working on side blotch lizards, set up these artificial rock piles, and he has cameras there and weather data, weather recording equipment. So he can, with a time-lapse camera, look to see whether the lizards are coming out even in the middle of winter. Pete also has some very large enclosures where he can monitor the behavior of the animals. And main reason I wanted to show this is these lizards are, during the summer months, living out here in the open areas and further out, and they migrate large distances to these rock walls here. And so that's where they're hibernating, but they're spending most of the year down on the flats. If you were trying to design a seasonal acclimation experiment and you didn't know that they had different habitats in the different seasons, you might set up your conditions based on what's going on here, out here. That'd be very different from what the animals are actually experiencing. So it's a trivially obvious point, but you really need to pay attention to the natural history of the organism if you're trying to recreate conditions that are ecologically relevant. You can also set up sort of microcosms like this. This is work from Kevin Roberts, Caroline Williams' lab at Berkeley. They were looking at overwinter survival of beetles as a function of snow cover, and they could set out these little enclosures, cover them with sand, and then look at them. So those kinds of studies are done. They're produce some really interesting results, but by far the most common methods are laboratory methods, as I said earlier. And let's look at just a typical sort of manipulation of body temperature simulating seasons. This is the simplest type of experiment. You have two groups of animals. One is exposed to both warm temperatures initially, and then the animals are temperature is shifted acutely to a winter condition. Let them go for a few weeks or a month and then make a comparison. There are variations on that theme where you step down, say once a week, change the temperature, or you do it every day. When I was getting started, the environmental chambers were pretty crude, and almost all of the studies were these acute shifts. Now with improved programming, you can do more complex types of patterns, but still the most common is an acute shift in temperature. Let's compare that to what actually happens in nature. This is a study of timber rattlesnakes where they implanted data loggers in snakes and released them into the field. And you can see the changes in body temperature of individual snakes over time. This is just obviously a regression line, not a snake. But there are obvious differences between what these snakes are experiencing and what laboratory experimenters subject animals in the lab to. There's daily variation, there's within day variation. It's a gradual change, and it's non-monotonic shift. And periodically you get some animals are coming out and basking and warming up. So these kinds, what happens to animals in nature, it's much, much slower and much more sort of stochastic than what's happening in laboratory experiments. Does that matter? I don't know, but I would suspect so. I mean, here the animals have 100 days as body temperature drops 10 degrees. And in a laboratory experiment, that 10-degree drop might take place over anywhere from 1 day to 2 weeks. So the animals have much longer to acclimate. That may mean they get a stronger acclimation response, or it could mean that the long exposures to low temperatures are deleterious. So in fact, this will be physiologically deleterious to them. We don't know because nobody's ever done a comparison. Most studies are also done with fixed temperatures. So you have a high temperature and a low temperature for summer and winter. Sometimes they'll put a daily cycle on it. But the question is, what are the body temperature distributions of animals in nature? I couldn't find a good example where people had actually done this. So I just simulated what one would expect using a program developed by Michael Carney and Warren Porter called NicheMapR. With that, you can take environmental data, and I took it from Fort Dry Lake in California, a desert area, put in some behavioral properties of the lizard, and you can simulate body temperature distributions during activity and inactivity. So activity is shown in red. Inactivity means that they were underground. And there's some striking patterns here. One is that the activity temperature of lizards does not change very much seasonally, even in winter. They're still getting pretty warm when they're out. The major shifts are, first of all, you see a bimodal distribution. So there's a daytime inactive modes. And it's the nighttime or the inactive temperatures where you're seeing huge shifts, not only in the mean, but also in the area under the curves. So if you're trying to set up a seasonal acclimation experiment, at least for an animal that is able to stay active in the winter, the activity temperature is not going to be changing. The inactivity one is a lot, and the areas under the curve. I know of only one study that's done this kind of a pattern, and only for 2 seasons. So it's an open question: does this kind of a body temperature profile over the course of a day make a difference? We don't know. I suspect it does. One way to find out what animals are doing, if you can't put a transmitter in them or a data logger, is to do what Bill Bradshaw, Pete Zaney, and Chris Holzapfel have done with pitcher plant mosquitoes. They find pitcher plants, they put in a data loggers. In those, in northern locality, southern locality, and do this over the seasons. And then you can see the profiles that these larval mosquitoes are experiencing in these pitcher plants. And they can use these then to program their environmental chambers to create realistic patterns of seasonal variation in temperature. For a pitcher plant mosquito, I think that's great. For a lizard, I'm not so sure, because a lizard, being a mobile critter, has the option to use its behavior. It's not just stuck in a pitcher plant, so it can move. And this raises the general question: what happens when you stop an animal for doing what it normally does? I'm going to use an example here from Ted Garland, who was a former postdoc in my lab. Ted has done some amazing studies on house mice and selecting on activity in wheel running activity. So he has some control lines that are running about 5,000 revolutions per day. And then he has some up-selected high runner lines where he selected on wheel running behavior. So if they ran a lot, he would use those for the next generation. And after 31 generations, these guys are running 3 times further per day than the control mice. So these are just hyper marathoners. So I wrote Ted and said, what happens if you take the running wheel away? You stop these animals from being able to do what they like to do, to run. And he says they show symptoms of depression and withdrawal, almost the same way that a human does if you take cigarettes away from a smoker or alcohol from an alcoholic. Let me show you another example of why I think behavioral, the fact that acclimation experiments take away behavior may lead to incorrect results. This is old work from Barry Sinervo, who's a grad student and postdoc in my lab. Barry, as part of his thesis work, did a study with finch lizards, hatchling finch lizards, where they had a thermal gradient, but the heat lamp was on for only a few hours a day, either 6, 9, or 12 hours. So the question is, if you give them an opportunity to be warm and active for 12 hours a day, do they actually use it? So here's Barry's data for California. So this is time since sunrise, or when the heat lamps came on. And you can see that they stay warm, even the ones that have 12 hours of light, until maybe the last time period when they drop their body temperatures a bit. But here, you give them a heat lamp for a long period of time, they use it. But look what happens when you go to Oregon lizards. They come out early in the morning, they'll start basking, but after a few hours, they move to the cooler end of the gradient, or they bury themselves in the sand. So they're not using, they're choosing not to take advantage of this heat lamp for the full length of time. So the question is, if you were doing a seasonal acclimation experiment and you forced Oregon animals to stay at 33 degrees for 12 hours per day, do you think that that's likely to induce a pathology to be harmful in the animals? I don't know, but I would bet that it does, because they are voluntarily selecting not to take advantage of those long time periods. So in other words, when you keep an animal from doing what it's being able to use the behavior that it would normally do, are you inducing a stress response? It's an open question. Yeah, so if you force them to be here, I think it'd be problematic. Here's another way to look at this. Imagine you have a patchy environment. So you have one animal in an area that's too cold, another where it's too hot, and over here, habitat that's just right temperature. In nature, they're going to move if they're aware of the fact that there's heterogeneity out there. But when you do an acclimation experiment, whoops, what you're doing is blocking access. So they're forced to experience cold or forced to experience warm temperatures. That will have a direct effect on their physiology, but I'm questioning whether or not the inability to use their behavior also induces a change. I think likely is. Let's move to photoperiod. When I started thinking about this project, I thought photoperiod is something that's easy to adjust in the laboratory. We know that photoperiod is a reliable cue of seasonality for animals and plants. And as Levins pointed out, it's a predictor of changes of other physiological factors that are important to organisms. Okay, so a traditional photoperiod experiment would do something like this, where you have animals exposed to long day, and then you acutely drop them some to a short day, let them go for a while, make a comparison. Or more recently with natural photoperiods and adjust those. That looks pretty good. So here is day length for Ford Dry Lake in California. Varies from about 9 to 14 hours, something like that over the course of the day. Question is, would a lizard at Ford Dry Lake experience a photoperiod remotely resembling the astronomical photoperiod? And I think not. So here, using NicheMapR, I've predicted the surface activity of these lizards. And you can see that in summer, they're above ground most of the time. But in winter, they're not experiencing a 9-hour photoperiod. They're experiencing one that's closer to 2 or 3. So the use of the astronomical photoperiod, I think, is valid only for an organism that spends 24 hours a day above ground, that's not shaded by trees or mountains. But for an animal that's mobile, like a lizard that can go into a burrow or something like that, astronomical photoperiods may be misleading. Does this matter? I have no idea. As far as I know, no one has ever looked to see whether this will have an impact on the observed acclimation shifts. Another complication is shown here, some paper with Miles and Pianka. This is a Kalahari lizard, and we're looking at body length of lizards across the seasons. This is winter. Ignore the red triangles. Those are animals that I dug up. But you can see in the autumn that you get a lot of adults. Oh, dashed line is demarcates adults and juveniles. Both adults and juveniles in autumn, but you go into winter, the adults are all hibernating, but the juveniles are not. And then in early spring, the adults come back out. That's not true for all lizards in the Kalahari. An arboreal skink, both adults or juveniles are active in the winter. But if you're programming photoperiod, you'd have to use a very different photoperiod for adults and juveniles in the Lacerdid, but that you could use the same photoperiod probably for the skink. But these kinds of dynamic factors, I think, are rarely considered. You really need to know the natural history of your organism. Gets even more complicated. If you remember that rock wall that I showed from Pete Zaney's enclosures, this is a close-up look in the middle of winter. This is January 28th. Air temperatures are close to freezing. And if you look here, there's some little blips. Let's blow that up. These are lizards packed in there. And many of them have just their heads out, but some of them are fully exposed. So these lizards are basking right in the middle of winter and in large congregations. During the activity seasons, they're very territorial and spread out, but to the winter, they come together and they're living in the same cracks and high densities. I mean, you can see about 7 or 8 lizards there just in this small little area. What are they doing? Are they foraging? Or are they just basking and getting warm and exposing themselves to solar radiation for vitamin D? We have no idea. But let's assume that they're not foraging, and I'll come back to that in a few minutes. Let's assume they're just coming out to bask, either for vitamin D or for physiological readjustments, which they can achieve by becoming warmer. If you do an acclimation experiment and you don't allow for this kind of periodic basking, you don't allow them to clear their metabolic byproducts, et cetera, I think it's very likely that the animal that you pull out at the end of the season is not going to be in as good condition as one that was able to intermittently bask. I have no evidence for that, but it just makes sense that if they're coming out, they're doing it for some physiological reason. And an acclimation experiment that doesn't allow for that may get incorrect results. So there are a lot of other issues that have been raised in the literature about photoperiods, matching photoperiod to thermal periods, adding twilight, spectral quality and intensity. All of these things are known to have important physiological effects. Point being that photoperiod is, I think, harder than it looks to get it that's sort of ecologically relevant. Certainly harder than I would have anticipated when I first started thinking about this. Let's turn to food. Two obvious issues here: how much to feed them in the laboratory in different seasons, and what to feed them. Most studies that we've looked at, Lauren and I, most of them provide ad lib food in the different seasons. Some, if they think the animals are not feeding in winter, they'll provide no food in that case. But most of these studies don't really comment about what's known about seasonal variation in feeding in nature. Here's an old study by good friend Ken Nagy working on side blotch lizards. Ken uses doubly labeled water, an isotopic technique that allows him to measure field metabolic rates of free-ranging lizards and also quantify the amount of energy they're taking in per day, joules per day. And you can see there's huge variation here over the course of the year, from 200, to over 600. So presumably then, if you're trying to set up an experiment like this, you should really vary the amounts of food that you're providing them at different times. In the Kalahari, we have data for an entire community of lizards. Most of them, these are food volumes. These are not joules per day, so it's a cruder technique. But if you look here in the winter, almost all of these animals are feeding, with the exception of these two species, which are hibernating. But in the Kalahari, then, animals are feeding. What about animals that are hibernating? Is it safe to assume that they are not feeding in winter? We don't see them above ground. Are they fasting? Here's a Kalahari gecko that I didn't see for almost 4 months in the Kalahari. But we were able to find some. We found, 18 in termite mounds. 17 of the 18 had food in their guts. Dug up 3 in the sand below fallen shrubs. None of them had food. Small sample size, but it's something. Anyway, for this gecko, they are inactive. They're not running around, but if they can find a termite mound in the area, they've got a thermal buffer. So cold is not going to be a problem. It's probably good, suitable temperature, and they have a source of food. But if you didn't know that, you might set up an acclimation experiment with no food, which would be inappropriate, at least for the ones that live in termite mounds or that seasonally move to these termite mounds. You don't see them in association with termite mounds during the summer. What about food type? This is an old study of chipmunks from Lloyd Tevis, and he found that you go into autumn, chipmunks start eating more seeds than they do at other seasons. This was of interest because it's known that seeds seeds are high in polyunsaturated fatty acids, and that animals that are cold tolerant typically have membrane lipids that are high in polyunsaturated fatty acids. It promotes membrane fluidity even in the cold. Jim Carnegie and Fritz Geisler did a study with these chipmunks manipulating fatty acid content and showed that diets high in these polyunsaturated fatty acids have big effects on their overwinter hibernation responses. Point being that if you're feeding in the lab animals rabbit chow for the entire year when you're doing a seasonal acclimation study, you may generate patterns that are biased relative to what would happen if you allowed the animals to change their diet as they do naturally. This can happen even in Drosophila. So study done where they took flies, put them in a petri dish and gave them access to, either yeast or plant foods. And if you acclimate them to warm temperatures, say 15 degrees and above, the flies go to the yeast. If you put them at about 12 or 13 degrees, suddenly many of them start feeding on plant material. So there's a voluntary shift here in what they're eating. What they did next was to rear flies on either yeast or plant food, put them in vials, stick them out on a roof in Dresden in October and look at survival. And the patterns here are stunning. If you give them standard fly food, yeast food, they die very quickly. If they had been reared and provided with plant food, they survive much longer. Probably because of the higher polyunsaturated fatty acids promoting membrane fluidity, which they documented. There have been studies done with Drosophila in outdoor cages, but many of those just using standard high yeast Drosophila food. One wonders whether those are realistic if the flies are able to change their diet in nature. Whether they do or not, I don't think has been established, but it would be really interesting to study. So the last area is something that was inspired by the last couple of years when we're all suffering through social distancing and lockdown. But when you take an animal out of the field and you put it into a cage, you are locking it down. You're preventing an animal from doing activities that it would normally do. And there's a series of things one can ask. Should the cage be just a simple little box, or do you have to have an enriched habitat, sand, cover, maybe something to climb on? Does that make a difference in what you see? Do you have animals raised in the winter, say, or summer, individually or in groups? In the case of Pete Zaney's Utah, they're solitary during most of the year, but they become very social in the winter when they're in those rock caves. Do you have conspecifics only? And in general, when you put an animal that's normally a mobile animal in a small cage, you're physically restraining it. Does that lead to deconditioning? It certainly would to us. Does it lead to stress? I suspect it might. I'll show you one example of sort of a complication that can occur from this issue of whether you have social groups or solitary animals. This is old work by Phil Riegel, working on a lizard called Clauberina. It's off the coast of California. This was an early study of temperature regulation in the laboratory. And Phil set up an operant conditioning setup where if a lizard came over here and sat on, sat on this, I'm going to try not to touch this. The lizard come over, sit on the micro switch, the heat lamp comes on, the lizard starts to warm up. But eventually it gets too hot and it has to move off the micro switch. The heat lamp goes off. The lizard wants to warm up again. It has to climb back up on the micro switch and do that. Okay, this is connected to an event recorder, so Phil could get long-term, over many months, record of how often the lizard used the heat lamp. So he did one experiment where he had two males. Each row here represents a single day. And if you look here, you can see little bips, and that indicates activity. Ignore the vertical lines. Those are just there for some other reason. During the first part of the experiment up here, Phil had two lizards in the cage, a dominant male and another male that was subordinate male. All of this thermoregulatory activity was done by the dominant male who prevented the subordinate male from getting access to the heat lamp. Here, Phil took out the subordinate male and noticed that the dominant male continues to thermoregulate for several days, but then he becomes a sloth and just stops doing anything at all. Phil then reintroduced the subordinate male here, and the dominant male now starts thermoregulating again. So the thermal profiles that these animals are experiencing, the metabolic rates, and undoubtedly stress responses that are going on here are dynamic and depend on the social context, and potentially could affect the seasonal acclimation experiment if you had an incorrect setup. So I've pointed out a number of issues that I think are problematic that are in the design of acclimation experiments. And I want to show what I think are some really positive approaches that set a very high standard. And for me, the gold standard would be work by Bill Bradshaw and Chris Holzapfel at Oregon, working on pitcher plant mosquitoes. Incidentally, their laboratory was selected in 2007 by the National Science Foundation as one of 10 laboratories in the United States in all fields that met the NSF goals of discovery science. So I'm not alone in loving their work. Oops. So you have these pitcher plants, you have mosquitoes growing in it, and they have beautifully controlled and have done this for decades in the laboratory with natural habitat, not vials, not cages, but natural habitats of pitcher plants. They have natural seasonal daily cycles of temperature, photoperiods with dusk-dawn transitions, and seasonally adjusted food. And they've just done amazing studies over the years. And I think this sets a really fine example of how you can do something in the laboratory that's ecologically relevant, but still maintains control. I think the opportunities for working in field enclosures are really starting to take off. Work here at Mouly, Elvira Bastion colleagues did a really nice study a few years ago with lizards in these enclosures, and they're able to manipulate temperature to some extent. So in these kinds of setups, which are ecologically, where the animals can move and use their behavior, I think a very effective way to look at seasonal acclimation. There's a long history, of course, of using phytotrons and ecotrons, ones that are developed now in France, where you can control many aspects of the physical environment in ways that I think offer spectacular opportunities for doing seasonal acclimation studies. Those limitations here, these are expensive to build and maintain, and relatively few people can get access to them. So to conclude, I'd say sort of two issues that, as has been pointed out many times in the literature, when we do a seasonal acclimation experiment, we're forcing animals to experience in conditions that may have little ecological relevance to what they would normally experience over the course of the season. obviously, the best way to improve that is to know the natural history of the organism that you're studying. Find out what kinds of temperatures and photoperiods they're experiencing, what foods they're getting, and so on. And try to make laboratory setups that are ecologically relevant. It's easy to criticize the literature and say, well, they shouldn't be using fixed acclimation or acute shifts. But it's also fair to say, is that level of complexity really necessary? Maybe an acute shift in temperature is good enough. Maybe using astronomical photoperiods are good enough. We don't know, because there are very few studies where people have taken animals from the field and looked at what their realized responses are and contrasted that with what you get from the laboratory. So we don't know how much complexity is necessary. But when I was thinking about this, I thought, wait a minute, you know, I've heard this debate between complexity and simplicity several times during my career, and thought back to this classic paper by Richard Levins, when he's talking about model building and contrasting approaches where you have these really detailed mathematical models where you measure everything and then make a specific prediction about something, as opposed to a very simple model with few parameters where you're trying to get just the essential features. And I think that's where we are. And I don't know where this will end up. How simple an experimental setup can we get away with and still get the essential features of seasonal acclimation? That, I think, is an open question, something that's going to be difficult to resolve, but hopefully we'll be able to do that. So thank you, and I'll be happy to take questions.
Thank you, Ray, for the great talk. So there's lots of people in the audience, so I'll just wait for questions to show up. Here's one. So just a second. Sylvain, I think you can now turn on your microphone if you want.
Yeah, can you hear me?
Yes, good to see you.
Hey, Ray, how are you?
Well, thank you. And
well, yeah, good. Yeah, thank you so much for your presentation. It's really nice to see also how to say. I remember last time we met several years ago that you mentioned very old papers that already mentioned stuff, but we were thinking that we were about to discover or have original ideas about something that actually has been thought for years or decades, and really it's refreshing in some ways to see how these old studies that already covered and started to initiate all this work, and I really love that. And speaking about seasonal acclimation, clearly, from what you say, there is a clear dependency on the available heterogeneity in microclimatic conditions that is available in the habitat of a given species. And I get the feeling that most of the research has been focusing on having the individual exposed to solar radiation versus under shade or in the ground. And I get the feeling also that it's not that simple and dichotomic. What do you think about that? If it's important to really include all the possible macroclimates within those that are exposed to solar radiation, and with all the gradual sort of gradient that you can get from all these habitats, macrohabitats that are exposed to solar radiations, and versus, of course, in the shade or into the soil, it's likely to be more homogeneous in time and space, I would say. What can you say about that to us?
there's a lot there.
Yeah.
Certainly what happens above ground and below ground is going to be very different in terms of the magnitude of fluctuations in temperature, photoperiod, and other physical factors. The issue of solar radiation, I don't know. My guess is that we see in many reptile studies that in winter, they will come out and bask, like Pete Zaney's studies or the rattlesnake study that I showed you. Mm-hmm. Even in very cold environments, they're doing something. And they're coming out for some physiological reason. We may not know what it is yet, but it's probably beneficial to them to bask, to be exposed to solar radiation, or to warm up, or both. And that just leads me to suspect that if you don't allow them to do that, you keep them in the dark for 3 or 4 months, simulating a hibernation, or maybe even 6 months, that you're going to be inducing pathologies into those animals. So I think it may be important. Whether it is or not, I don't know. Maybe it makes a 2% difference in their performance, and it's something we can just ignore. So I think it's an open question.
Yeah. If I may ask just another question while I'm here. Do you think it's the same? I mean, if you target a body temperature of, say, 30 degrees, do you think it's similar from a physiological point of view to put the organism at 30 degrees in the air, or to allow the organism to bask into the sun to reach 30 degrees? You see what I mean?
So whether you force an animal to do it or you let it make its choice.
Yeah, I mean, in both cases, you impose these 30 degrees, but in one case it's by air temperature, and in both cases it's from radiation.
Yeah. It depends. The body temperature will be the same. The primary difference is that the one that's exposed to solar radiation is exposed to solar radiation, which can have both beneficial and detrimental effects. It may promote vitamin D synthesis, but high levels of ultraviolet can also be physiologically damaging. So I think there's a potential that solar radiation and air temperature both can lead to the same body temperature, but you may have somewhat different physiological outcomes.
Okay, we should move on to the next question. Thanks. So Anne Charmentier had a question too.
Yes, hi, thank you very much. I enjoyed your talk very much. I don't work on lizards, I work on birds, but the general message of paying attention to experimental details to make sure that the results we obtain in the lab make sense biologically really resonates with a whole set of questions that I've been asking myself recently because of experiments that we're going to conduct on a bird species. And my question was to try and solve these questions. My question is basically, what is the best way to measure the level of complexity that is necessary? So would you say, for example, that comparing variation expressed in the wild with variation expressed in the lab is a good way to see whether natural variation is well represented in the lab? Or do you have other measures in mind as to how we can, yes, I guess, compare the responses that we get in the lab and in the wild and make sure that we reach the right level of complexity?
Well, I think your suggestion is a very good one. Just compare the animals from the field versus the ones exposed in the laboratory conditions. There are a few studies that have done this. Sometimes the results are similar. Sometimes they're a bit different. And so there's very likely to be no simple answer that applies to all organisms and all environments. And I think we'd just like to know how close can we get with a simple laboratory experiment, and then how much complexity do we have to add?
So my feeling is that we will go about doing this by trials and errors, right? Because when you build your experiment, you're going to try and add a certain level of complexity. But then if you end up having results that show responses that are too far away from what you expect in a natural system, then you're going to have to try and do better. But it feels like it's difficult to target the right level of complexity the first time you try this kind of experiment.
I suspect it'll be hard even the second and third time, just because there's so many different things. But I guess my feeling is the best place to start would be to know the natural history of the organism. What are they doing? Are they hibernating? Are they, what are they doing in the different seasons? How does the social environment change? If you have something like a lizard that can burrow or go underground, it's going to be very different from an animal that has to spend 24 hours outside the way a bird might be. But if you know the natural history, if you know that there's seasonal changes in diet, seasonal changes in body temperature or moisture content, if you know that, then you can select those environmental variables that are most likely to be different over the course of seasons. So I think I'd always just start with the natural history and say, what are they doing? And if the diet is staying the same over the season, so that may be something you don't have to worry about. But if it's like these chipmunks where they're switching to a diet that's high in polyunsaturated fatty acids, that may be something that you need to include. And certainly there's enough known from the physiological literature to suggest that that is an important factor in things like cold tolerance and seasonal metabolic responses. So I think the advice would be work as what you can from the natural history. The obvious challenge is that often we don't know much about the natural history of animals over all seasons.
Thank you.
Because most of us are teaching or writing grant proposals or doing something like that in winter. We're not going out and looking to see what animals are doing or exposed to.
All right, thanks. We're going to move to the next person, Cameron Gallenbar. So we have audience coming from different places.
Yeah.
Cameron, can you hear us? Can you turn on your mic?
Yes, can you hear me?
Yes.
Yes, I can.
Hi, Ray. How's it going?
Good.
Greetings from Norway, where it's actually quite a nice sunny day. If I was a lizard here, I think I'd be out basking out in the sun. So love the talk, a lot to think about. My question is that the whole time you were talking, my mind went back to the debates and arguments about the beneficial acclimation hypothesis. And I'm sure maybe we're inspired partly to ask these questions because of that. I suspect that a lot of the examples where people were arguing that acclimation was not adaptive or not beneficial were probably artifacts of the acclimation conditions that they kept the organisms under, and saw what were probably stress responses, and interpreted them as, these maladaptive, non-adaptive responses, when in fact, had they done a better job of a proper acclimation treatment, or even a longer acclimation treatment, they would have got very different results. I don't know. What are your thoughts on that?
It makes sense to me that could be a contributing factor to some of the anomalous results. To the extent that it is, I just don't know. But it's certainly a possibility, and I think it's a question that's worth addressing.
As you were giving your talk, I was pulling up some of these old papers by Laura and others and looking at their acclimation treatments and thinking, yeah, this is all stress. So,
well, I mean, when we did some experiments on Drosophila, we manipulated father's temperature, mother's temperature, developmental temperature, and adult temperature. But we used fixed temperature regimes, and we didn't change photoperiod. We used a fixed photoperiod throughout. Was that the results we have bear any resemblance to what would happen? Later work has shown that photoperiod has sometimes has the bigger response of things like heat tolerance than temperature acclimation. We held photoperiod constant. So, I mean, I'm not going to do that experiment again. It was really difficult. But I wouldn't be surprised that many of the results that we see are anomalous because of the laboratory conditions. I hope that's not the case. Yeah, because we want the data available that we do have to have some level of realism. I think
Sure.
it'd be more realistic if the acclimation treatments were ecologically relevant.
Yeah.
But it's an open question how much of this is a problem. Incidentally, I should say, and I should have confessed this right at the beginning, that I have never really done a seasonal acclimation experiment. When I was getting started in biology, I thought, when I was looking towards my career, I was saying, you should never criticize a field where you haven't played the game. And so I'm doing that. I'm being critical of a field that I've never directly been involved in. I'm doing it because I think these are important issues that need to be addressed, even if I myself haven't done them or won't be able to do them because of where I am in the stage in my career. But I hope other people will.
Yeah, I think
good, thanks.
everyone will agree that this is useful, I think. So we have another question by Wilco, perhaps?
Yeah, can you hear me?
Yes.
Yes.
Thank you. So great talk. I really enjoyed it.
Thank you.
I don't know, maybe I don't have a real question, but I thought about this, that maybe a quick way to gauge how important these more ecologically relevant conditions are could be to make a comparison between studies where they use these, what you call the golden standard. So they basically have wild captured animals and see what are the effect sizes or maybe the directions of the things that they find, and compare that to studies that have used these very artificial conditions in the lab. Do you know if somebody has already done that, or
Not on any kind of a comprehensive way. There are single studies where people have done field work and lab work and commented about one or two traits. To my knowledge, no one has ever done a systematic review. And I know of a few papers. The manuscript that Lauren and I are working on has a few examples that we know of. I'm sure that there are others out there, but it would certainly be a worthwhile project to do exactly what you're suggesting, where somebody has worked lab in the field, look at the effect sizes and look to see how similar they are.
Yeah.
I think that's the only way we'll ever really know.
Well, the other way is to use experiments, I guess, but this might be a quick way, especially if you also combine it with what Cameron Gallenborg said, to see if it's fitting with the beneficial acclimation or not, and then you could score those studies whether or not they support that. And well, one expectation would be, of course, that the wild captured animals show more often support of that.
That may be the case, but consider if you're working in an animal, say at the northern limit of its range, where it's exposed to a very long winter. It might well be that what you see is a physiologically damage for long exposure to extreme cold. So yeah, you could be measuring a deleterious effect there that's swamping any beneficial acclimation.
Yeah, that's true.
So Sylvain Ainsbourg seemed to have a reaction to these comments.
Oh, yeah, well, it was just a thought in line with what Wilco was just saying, that another way would be also, another way, and a parallel way would be also to compare the seasonal acclimation effect size of aquatic versus terrestrial ectotherms, especially given that aquatic ectotherms are thought to live in relatively more homogeneous thermal environments compared to terrestrial animals. Fishes cannot bask like a reptile in the rock, you know. So I was thinking with Lauren, if you thought about comparing directly this effect of seasonal acclimation of aquatic and terrestrial, and what would you expect in terms of amplitude of this seasonal acclimation effect between the two categories?
It's a good question. I mean, it's an interesting one, given the differences in that you see in the physical environments of aquatic and terrestrial systems. We haven't done that. I don't know anyone who has. There are studies looking at comparing aquatic and terrestrial animals. I've not been involved in those, and I don't really have a feeling for the aquatic environment. Yeah, there was another example, but I'm sorry, lost my train of thought on that one.
Thanks, Ray. Okay, thanks. Luis Miguel, le micro il est coupé.
Thanks. So I was saying we can move on to the question by Tobias. Hi, Tobias.
Hi, Louise. Hi, Ray. Great. You can hear me okay, I hope.
Yes.
Great to see so many lizards. It made my day. Thank you so much for that. So, you know, as a lizard biologist, I think a little bit about this every now and then in the context of encountering really long spells of bad weather in some environments. So when you wonder if the risk of being exposed to several weeks of rain and cold will then make those animals require longer periods to acclimate, because otherwise they will sort of misacclimate when they don't need to do that in terms of the seasonality. And if we know anything about that, has that been studied? That if there are differences here between lizards that live in places where they can encounter bad weather for weeks, if they require longer periods to acclimate than lizards that live in places with more reliable activity conditions.
I can't think of an example right offhand. The closest might be an old study by Steve Palumbi on sponges in the intertidal. And he did this decades ago. It was a beautiful study of sponges that lived in areas on the outer coast of Washington where they're routinely getting smashed by waves, and other ones that lived inland or in a protected area where there was very little bashing by waves. And the sponges that were in the exposed intertidal area had lots of spicules and small holes, and they were very robust compared to the ones that were inland and protected. Steve cut pieces of sponge, transplanted them to the two environments. And what he found was that if you take a sponge from a protected area and you put it in a rough area, that the sponges develop the morphology of the windswept areas very quickly. If you took the ones from the coast and moved them inland, they were much, much slower to respond. And Steve's idea was that it had to do with the predictability. Once you get smashed by waves, very likely you're going to get smashed again. But if you take away that stress, like you take them from the outer coast, move them to the protected inner coast, and suddenly they're not getting smashed by waves, it's unlikely that that's likely to be just a lull period. And so he was proposing that the timing, the rapidity of the shift in morphology had to do with the predictability of an environmental stress. That's about the closest I can think of to someone who's done that. I'm sure there are other examples out there.
It could potentially be a way of also getting at how important this is, right? Because if if these things do differ, say within the same species of a lizard that lives in the mountains or somewhere down in the lowland where the summers are much more reliable, if those differences exist, then chances are that this is going to be, we're going to mess about a lot with the way that we do lab experiments. Whereas if it's not so important, then maybe they are a little bit more robust to also the differences that we do to them in the lab.
Yeah, I was just trying to think if anybody has done, in typical laboratory experiments, you transfer animals to a new condition and then you leave them there. And I can't think of experiments where people have changed the conditions once you set up an acclimation. Typically, high temperature, low temperature, leave them there and measure them, and not doing periodic manipulations like might happen in an unpredictable environment. Somebody must have done that, but I'm not thinking of any example right offhand. Cam might know of one.
Thanks, that's great. Thank you very much.
All right, I don't know if there are other questions, but I think I had one myself or comment, and I don't know. The idea is that, of course, it's very difficult to simulate all this environmental complexity in the lab. And my understanding had always been that this, you can hear me well?
Yes.
Yeah, my understanding had always been that these thermal tolerance or performance experiments were not really, people know they're not really realistic, but it's thought to be some proxy for what's happening, a little bit in the philosophy of Arnold's paper about performance, where you use performance as a proxy for fitness because it's difficult to measure fitness in the lab. And so you use the performance as a proxy, and then you use this to compute selection gradient, for instance. So what I kept wondering during your talk is that would there be a way to do some kind of bridge experiment to relate what you measure in the lab to what happens in the field, and then still doing simpler things in the lab and trying to connect, do some what I call bridge experiments that try to see how relevant they are to the field, but still keeping, because most people, I guess, would not be able to have this level of complexity to simulate the real thing. But what they would want to know is when they're using a somewhat unrealistic setting, to what extent it's going to capture part of what's happening in the wild. So I don't know if you've been thinking a bit about this, how experiments that try to connect the two or try to correlate them to be able to.
Well, if I understand what you're saying, what comes to mind is work that was done by Ari Hoffman, Volker Loschka, and colleagues with Drosophila, where they would rear flies in the laboratory, say under different temperatures, and then release them into the field at different seasons and see whether or not the flies that had been reared at high temperatures survive better in the summer, and conversely, cold ones in the winter.
Or something like, for instance, flies that undergo stronger effect of heat shock, for instance, do they also survive less? Or these genotypes, do they also survive less? Not just because they have been acclimated in the lab, but just because these genotypes that tend to resist less well to heat shock, they also perform less well and survive less well, something like that.
Yeah, absolutely. And I think Martin Fieder's group has done some things along the lines with fruit flies and heat shock response. But yeah, I think those are approaches that will, I think they're useful. I'm not sure that they get directly to the question. So let's say you take flies, you acclimate them high and low temperatures, take them into the field, and you show that they survive better. High temperature ones survive better in summer. Does that mean that necessarily that natural flies in summer have elevated heat tolerance? Probably, but not necessarily. I mean, it could be that flies in nature in summer are heat stressed, and they actually have lower heat tolerance because of the cumulative stress of high temperatures. But I like the idea of this kind of an experiment where you can do something in the lab, test it in the field, and see if it's giving you results that are equivalent to what's happening in nature and having some adaptive pattern.
Okay, so I see there is a question by Fred Mary, so perhaps we can take it quickly. Fred, can you turn on your mic? About social context? Yes, no, can't hear you. All right. Then if not, I don't know if other Patricia, yes, Fred, can you turn on your mic then?
Hey.
Patricia, you had another comment or question perhaps?
No, it's okay. Thank you very much. It was very interesting, and all the questions have been already asked.
Okay, so I guess people who still have questions can write to Ray and send him an email. I think I would like to thank Ray again for a very dense and thought-provoking talk. Then for the rest of the audience, I guess, well, we'll meet some other time for the webinars, and we'll keep everyone in the loop for the next one. So have a nice day.
Okay.
Bye-bye.
Thank you all.
Thank you, Ray. Bye-bye.
Bye.